Right, so thank you for the invitation. I want to take a couple of slides to introduce myself, introduce the general context of the work, and then I'm going to talk a little bit about the work we do in unconsciousness, and I'll explain what that means, and sort of try to explain how we got into the psychedelics work from unconsciousness. So for about 10 minutes, I will run the two in parallel, and then we'll go properly into psychedelics. So my name is Emmanuel Stamatakis, as you already know. So let's start with telling you what I'm interested in, and the kind of questions that I deal with mostly revolve around brain injury, especially severe brain injury. So severe brain injury that leads to unconsciousness. So people in coma, or what we call minimally conscious state. And we try to understand what happens to the brain of those people and why they cannot wake up. And at the same time, we run experiments with anesthetic drugs like propofol, which are routinely used in operations every day in hospitals. So we make people unconscious. We have people who are unconscious because of an illness, and we look at what happens to brains with psychedelics too. So a couple of very, very basic slides on what I mean when I talk about connectivity. So the tool of my research is an MRI scanner, and with an MRI, especially, sorry, I'm trying to start the timing of this so I know how far I'm going to talk. So what do we do with an MRI scanner? We take scans of roughly how oxygen moves around in the brain. And we put all those scans together. So we design experiments like this very simple one here. So we have an on and off condition, an on and off condition. So a person in the scanner does this for about 30 seconds, and then they stop doing it for 30 seconds, and then we subtract the two, and we get these colorful pictures that you see here. So these parts of the brain, primary motor areas, motor areas, responded to when we were doing this, this action here. And beyond that, we can ask more complicated questions, such as which parts of the brain communicate with other parts of the brain while we're doing this, while we're doing something, we're engaging with the stimulus. So that's what we call connectivity. And connectivity questions don't have to be one-to-one or one to other parts of the brain. They can also be answered by building matrices like this, where we look simultaneously what happens, how each part of the brain connects to every other part of the brain. So we can build matrices like this. These are correlations, basically, between two time series that we extracted from two different parts of the brain. We can use different atlases to break the to separate parts of the brain. And we then, after we build these matrices, we can calculate properties, and we'll talk a little bit more about these properties and how useful they are a little further down the talk. Now, a lot of the work that has been done with fMRI concerns tasks, putting people in the scanner and asking them to do something. However, 15 years ago, we discovered a little bit more than that, actually. We discovered that even when we ask someone, a volunteer or a patient, to lie in the scanner for about 5 minutes to half an hour, we see these patterns that you see again in the colorful blobs there. I say blobs because Morten said blobology earlier, and this is what he meant. So we see these patterns that we call networks. So in other words, all the areas that you see here co-activate when we are in the scanner doing nothing for 5 minutes. So they fluctuate in activity, they co-fluctuate in activity. And we gave them names because we know these posterior parts of the brain in the occipital cortex have to do process visual stimuli, the same for auditory stimuli, executive function here, and so on and so forth. And we can see, I mean, the reason these things have become so popular is because they tell us something about brain architecture, how the brain is organized. And they also very, very useful when we have patients that they cannot do tasks. Not every patient you can put in a scanner and say, can you move your, can you make your index finger to touch your thumb? Or can you engage with these regular and irregular verbs and press a button as a response? So resting state networks, which are acquired from resting scans, can convey a lot of information. And a lot of the talk that I'm going to do today has to do with resting state networks. But to try and persuade you a little bit further that they are useful, I'm showing you here somebody, again, doing this task. And this is, if we ask the question with what we call a connectivity analysis, tell me which parts of the brain relate to M1, primary motor area, while you're doing this task. The reddish areas are what you're going to get as an answer. And we can ask the same question when the person is not doing anything, and the orange areas there is what you're going to get as an answer. So you see there's a huge overlap. The areas engage more when you're actually doing the task than when you're not doing the task, but there's still the same network is there. And to persuade you even further, Cole and Bassett put together 7 tasks. And so here we see a matrix that's been constructed from putting 7 tasks together and 56 minutes of resting state. So you can see they're not identical, but they're very, very similar. In fact, they correlate by 0.9%. Now, when this whole discussion about resting network started, there was one network that we had not encountered before. And this was called, then the name was given the default mode network. And we had not seen this network before. And we noted that it is very, very prominent in this resting condition. So, of course, the big question was, what does it do? Does it do these parts of the brain that you see down here and up here, are they important for thinking what you're going to shop from the supermarket after you finish with your scanning? Or are they important for planning your future? Or are they important for thinking about your past? All of the above, it seems. But some people suggested that that's the only thing they do. That's the only thing they do. You have half of your brain which you use to basically ruminate and plan and such stuff. And we published a lot of work to show that is not the case. How can you be conscious if you don't have all the information that you have learned so far? If you have all your knowledge disappears, can you be conscious? So, we kind of published a few papers, but this picture is a good illustration of how the DMN, so the default mode network you can see down here, and each of these strands represents how a part of the brain relates to another part of the brain. And here you have a working memory task that gets increasingly more difficult, so very, very difficult at the end here, where you have to remember, you see single characters on the screen, and you have to remember whether 3 characters back is the same as the one you're seeing now. And you can see that the more difficult the task becomes, the more you get these strands of the DMN relating to these other frontal parietal areas that are essential for the task. And we took this a little bit further, and we showed that the posterior part of the DMN here, and this is work that Diane Liu did, the posterior part of the DMN here is very important for integrating, for bringing information together. And all of this, if you believe in models such as predictive coding, you understand the usefulness of the default mode network and how it facilitates integrating information. And Diane showed that actually the posterior part of the DMN here is important in deciding whether you want to access more information from the environment or whether you want to access stored information in order to interpret what's happening from moment to moment. And this relationship changes depending on how familiar you are with your environment. Now, the other thing that I quickly skipped is a German psychiatrist called Georg Northoff is interested very much in the self and the idea of the self and how is that represented in the brain. So I'll give you an example of an experiment that can involve showing the person in the scanner a picture of a toothbrush, and you can ask them, think you're doing something with this toothbrush, and think your best friend is doing something with this toothbrush, and think the queen is doing something with the toothbrush. So you have degrees of separation. Now, the closer you get to yourself, the more these medial areas that are marked here with these dots and colorful shapes are involved. So each of these, the experiment I gave is an example, but each of these is a different experiment that activates those medial areas that heavily overlap with this default mode network when you do tasks that relate to yourself. The other reason why we got quite interested in the default mode network is because it has been written, there are numerous papers now that talk about involvement in neuropsychiatric and neurodegenerative diseases. So you have autism, hyperactive medial frontal cortex, prefrontal cortex. You have amyloid plaque deposition overlapping heavily with where the default mode network is. You have implications in schizophrenia. You have the default mode network being hyper-connected when you have depression. You have the default mode network being connected in lower than normal in PTSD. And both PTSD and depression are really important to us from a traumatic brain injury perspective, because a lot of those patients suffer from those conditions after the after their injury. So Ioannis Papas did an experiment where he used a model where a hidden Markov model, in fact, and this allows you to split the brain into states. You can define them any which way you want. In this case, he used the principal component analysis, and he found that these two states relate to depression score. Now, these two states are not identical to the default mode network. However, they relate heavily to areas that we call default mode network. And what he found is that going along with this idea of rumination in depression and the inability to get out of that state, he found that in terms of probability, you switch between those two states most frequently, and it's kind of very, very difficult to get out of that. And these big arrows, these thick arrows signify that it's more likely that you will stay in here forever and ever and ever and ever, and it's more difficult to get out of this situation. The other reason that I was interested in the default mode network is that it has been shown that the more unconscious you become, the more disconnected your default mode network is. So whether you look here at the posterior part of the DMN or the frontal part of the DMN or the parietal, inferior parietal areas of the DMN, you see that as you go from healthy controls, I can't read that actually, can you read that up there? Okay, so you go minimally conscious, a vegetative state, and then you have coma patients. You see in whichever part of the DMN we look, it becomes increasingly more disconnected as you become more unconscious. And theories, now trying to put this with theories of consciousness, that such as those proposed by Giulio Tononi, who says that for the brain to be conscious, it's really important to be able to put information together, so integrate information, but also keep information apart, so segregate information. Information that comes from all the sensory cortices have to be kept apart up to a stage where it needs to be integrated and construct concepts, if you want. To this end, Andrea, who will speak about another piece of research later, looked at data from people who have received anesthesia and sedation in this case, actually the same drug, propofol, and he looked at people who have disorders of consciousness, so they can't communicate fully, and he did that with an index, by using an index of connectivity. So he looked at every single voxel in the brain and to compute how connected this voxel was to any other voxel. And he also looked at diversity, a proxy for how segregated different voxels in the brain are. And then he overlapped the two, and he found this area in the posterior DMN, in the posterior cingulate, and precuneus areas. It spans both of these areas. And then he did the same for patients in disorders of consciousness, and he put the two together, and the overlap between everything was in the posterior DMN, telling us that posterior DMN is important for consciousness. Now, if you remember earlier, I talked about constructing more complex relationships than one-to-one relationships, and making up these matrices and calculating properties. And one of those properties that has been discussed a lot in the last 10 years in the context of brain organization is small worldness. Now, small worldness means an arrangement like this. So densely connected with each other's neighbors. So each of these could be us in a social network, or could be a part of the brain and its relationship in terms of activity with the rest of the brain. So here we have each of those areas, nodes, are connected strongly on a local level, and but they also collected on a global level with other areas. And the balance between this integration and segregation is called small worldness. It has to do with efficiency of organization, this balance between local and global connectivity. Now, the important thing is that we know the levels of organization, this balance, we know what the value for this balance is in a conscious brain, and we know we can test whether in unconscious people, it goes up or goes down. Andrea went one step further than this and calculated, given the theories of consciousness and the importance they give to integration and segregation, Andrea calculated two states. They calculated states in the brain that are very integrated, so instances, if you want, out of 5 minutes, that the brain does very highly integrative work, and when it does when it's important, segregation is important. And he found that in unconsciousness, what is really important, the small worldness is reduced at the periods of integration. So when you're trying to put information together, or in the parts of the brain that information is put together, small worldness decreases with anesthesia when you're unconscious. And I've given you some evidence now about the importance of the posterior medial areas in the brain in terms of consciousness. Now, at the same time that we were doing this work on consciousness, the team at Imperial was doing a lot of interesting work on what psychedelic substances like LSD and psilocybin do to your brain, well, how they change function and functionality in the brain. And what they found at the same time, and the reason I went all this long introduction about the DMN, is that integrity in the posterior, especially in the posterior parts of the DMN, you can see it here specifically, is reduced with LSD and changes with psilocybin. So we have in the same part of the brain changes that we found that is important for consciousness. And what happens is it changes in the opposite direction from the one that we showed for unconsciousness. So furthermore, remember the picture with lots of dots in the midline of the brain, self-related processing. Furthermore, there is a relationship between the integrity of this posterior part of the default network and the ratings of ego dissolution that people describe after they've taken LSD or psilocybin. Oh, here's a picture that I was. So you can see that in this part of the brain, we have coincidence between LSD, psilocybin, propofol, which is an anesthetic agent, and people with disorders of consciousness. Now, Robin Carhart-Harris at Imperial and all the team there also came up with this idea that has now moved on a little bit, expanded rather, that in order to measure and understand what consciousness is, we need to look at entropy and complexity in the brain. We have already started doing this work in unconsciousness, in patients and healthy people. So we thought, the measurements we're doing were down here in reduced consciousness, in a case of, in a system that is subcritical. So we thought, well, what we can do is test our methods initially and see whether we'll get values that are up here. So the way we got into psychedelic research is initially trying to confirm that our methods worked, and these models led us to correct predictions. So here then we have a continuum where we have a critical zone that the brain works in normal everyday kind of way. And then we have higher entropy where we have things like psychedelic states, we have REM sleep, we have psychosis, and we have low entropy where we have anesthesia, we have unconsciousness, we have either psychiatric conditions like OCD and addiction. And Ioannis, again, was the first person he looked at complexity of connectivity in the brain. And this was never published, but it was kind of the first result from the lab. So I quite like showing it. So he showed that when you look at the complexity of connections, you start with disorders of consciousness down here. So very, very small complexity. You have sedation, where you've given propofol. You have awake, healthy people, and you have psychedelics up here. So you see an increase in complexity, confirming the theoretical models that existing. And of course, we did this with more, in a slightly different way. Theories from physics suggest and have shown that the more near criticality a system is, in this case the brain, the more fractal it becomes. So Thomas suggested, why don't we look at fractal dimension in these brains of these patients who have disorders of consciousness? And the difference here is these people appear to be awake, but they don't really communicate, minimally conscious, and we have also vegetative state patients down here. So you can see as you go from healthy awake people to minimally conscious to vegetative state, the fractal dimension of the networks, the brain networks constructed in the way I talked about earlier, decreases. So the important thing here for us who study brain injury and disorders of consciousness is that this measure seems to be able to differentiate between minimally conscious and vegetative state, which can be quite useful in terms of making clinical decisions. Now, Thomas then went on to do the same thing with people who have taken psychedelics. So here's a brain of an awake person who has taken no drugs whatsoever, whether anesthetic or psychedelics. And here's how connected the brain of a person who has taken LSD. So you can see those matrices I was talking about earlier up here. You can see that this is a lot more complex than this. And if you try to quantify it, I should say here that all of our data on psychedelics have been shared by the Imperial group. When you try to quantify this, you can see that you get a very significant increase in fractal dimension in the brains of people that have taken LSD, and the same with psilocybin, although not as strong. It's still significant, but not as strong. So we have some sort of increased criticality on the brains of people who have taken psychedelics. And if you look, and this was looking at the whole network, and this is when you look at time series from individual parts of the brain, and then you average them, you get exactly the same result, except the psilocybin result is not significant. When we look at complexity, and in this case, Lempel-Zip complexity, at this stage, this kind of measures were used with EEG data, which is richer in terms of temporal resolution. You see that, again, the complexity of brain function increases when you take psychedelics and decreases when you take sedative or anesthetic agents. Now, Andrea came along again and said, I want to extend a little bit the work that I've done on anesthetics. So this time on segregation and integration. So it's a complex slide, I know, but I will direct you to the areas in the red rectangle. So in the first instance, what Andrea looked at was to see how the structural makeup of the brain differs from the functional connectivity in the brain. So you have the scaffolding, you have the white matter, you have the axons that connect different parts of the brain. So you're asking the question, if you construct a network, a matrix like the ones I've showed you, how does it differ from the functional one? So if you put the two together, the data there in terms of anesthesia and unconsciousness was that if you are unconscious, the two look very similar. So you don't have any richness in the functional connectome, as they're called, these matrices. So you have a functional connectome that's very, very similar to the structural connectome when you're unconscious. But again, exactly the opposite thing happens when you have taken psychedelics. So the psychedelics seem to increase the complexity in the brain, and this with very many different ways of showing it. The additional thing that Andrea found was that although in the context of consciousness, we saw that the stage that the integrated state, if you want, in the brain is more relevant. However, in the case of psychedelics, is where you see all these differences in the segregated states. So there isn't as much, the more important phase in this kind of two phases, it seems to be the segregated part rather than the integrated part, which we saw changes for propofol and people in disorders of consciousness. So we tested with yet another method the complexity. The results remained as they were. So we see increased complexity with psychedelics, and we also showed decreased functional correlations. Now, from the resting state networks that I showed you earlier, one of the big take-home messages was that the brain is permanently, especially at low frequencies, in this anticorrelated state. So if you're resting, if you're urged by a researcher to lie in the scanner and think of nothing in particular, you have the full mode network that is strongly anticorrelated with these executive frontoparietal networks. And we found that when you take propofol to be anesthetized, or when you are a patient with disorders of consciousness, these anticorrelations are reduced. And this time we saw exactly the same thing with LSD. So the anticorrelations, this kind of tension in the brain seems to be reduced when you take LSD. And we calculated small worldness here again, a property of these graphs, and found that it relates the difference in small worldness between a state of being awake and when fully awake without having taken any drugs, and when you take LSD, relates to all these things that people report after they've taken psychedelic drugs. So things like increasing complex imagery, ego dissolution that we already discussed earlier, and this blissful feeling. Now, this work was done, as I said, in a broader context of understanding consciousness and consciousness, understanding whether the methods we developed work in the way we expected them. But at the same time, of course, psychedelics are being enjoying so much attention now because we see them as agents that can help people with psychiatric problems which are sometimes resistant to drugs. And there is, as you can see here, there is increase in the number of registered clinical trials that are happening. And that's the kind of direction that we went after we developed all these measures. So in this particular case, we wanted to bridge data from the brainstem with all these cortical networks. This is all in the context of work that we did, again, with unconsciousness and found out that dopamine and the dopaminergic system is really, really important when it seems to be disconnecting when you are unconscious. So we try to do the same thing, and as I said, we focused on a part of the brain called the brainstem that you can see here. And Lenny, who's also here in the audience, did this work and looked at two strongly serotonergic nuclei in the brainstem called the raphe nuclei, the medial and the dorsal raphe. And what he found was that when you look at the resting state connectivity of this raphe nuclei, which are major sources of serotonin in the brain, you can see that they relate to that same posterior area of the DMN, the default network that we discussed earlier. So how does this relationship go? So we see that if we take it a step further and say, how does this area that we found connect to the rest of the default network? We find out that the more connected the median raphe nucleus is to this posterior part of the DMN in the precuneus PCC, the more this area relates to the rest of the DMN. So we see some kind of serotonergic modulation of the DMN by the median raphe in the brainstem. The same was noted for the dorsal raphe, but not to the same extent. So now we go to the really, really interesting and important study that was published last year in the, if I'm not mistaken, 2021 in the New England Journal of Medicine, which showed that if you compare psilocybin treatment with escitalopram treatment, which is a really commonly used SSRI, what you find is that psilocybin may work slightly better. And there was no imaging data that was published in this original study. And we asked the people at Imperial who collected the data if we can use the data and use the same kind of technique that we used to do the discovery about the dopaminergic system in unconsciousness. And we tested all possible nuclei that we have ROIs for in the brainstem, so various neurotransmitter systems. And what we found was that the greater the median raphe, which is shown here in green, connectivity, pre-treatment, so the greater the connectivity, the co-fluctuation between these two areas before treatment, the smaller the depression score is. So if you look at this, so now we're talking about some kind of diagnostic marker of people with depression. So we're saying that the more connected these areas are, so the more connected they are down here, the least depressed the people in this population are, okay? So then Lenny took it one step further and showed that the functional connectivity, so the relationship in terms of activity between these two areas correlates with the difference in depression score. So the more impaired the connectivity before the trial, before you took the drug, the greater the strength of the treatment response. So we're going from diagnosis to prognosis and perhaps treatment monitoring, because the third relationship that we found was that the strength of improvement in QIDS, which is the depression score assessment, was associated with higher median raphe precuneus connectivity. Excuse me. So a little bit of a summary then. A ZZ escaped there somehow. That is the wise part of the summary. So activity, what we find then is that activity in the brain on psychedelics is more complex than normal. we found that LSD makes activity in the brain a lot richer than it was, and it makes it a lot more different than the scaffolding, the matrix that we form by just looking at the scaffolding in the brain, the white matter connections. We found that increased small wellness, a graph property, predicts LSD-induced ego dissolution, and we found that LSD is important for segregated states rather than integrated states, and it has the more effect in those states, that is. And we found that the results in a major depressive disorder cohort provide empirical evidence in favor of psychedelic-induced serotonergic system reset for therapeutic effects. But I mean, take that with the last one, especially with a pinch of salt. A lot more work needs to be done on that front. But thank you for your attention, and thank you to all the people who were involved in this work.
I'm sorry, when you presented your functional data, I read it. There was one outlier of the control group who always looked at their own, whatever the psychedelic was. You have an theory to cover. The rest of the data was really tight, and then.
Sorry, which one is that? Was it in the propofol or?
Yeah, and there's three sets. Like, what's going on with their website?
Oh, yeah. Maybe they went as awake as I described. No, I'm sorry, I don't. But yeah, how tight everything is here, and then yeah, that person down there. Maybe they maybe they taken something before they before they came to be scanned.
I mean, is that kind of? I'm just wondering whether any of this is genetic, right? And then you get kind of the differences.
I mean, we don't have genetic data on those people, but what we have maps of gene expression from atlases that have been published, and we're looking at that now. But so maybe, I mean, yeah, maybe we're not in a position to provide conclusions about individuals, but we're getting in that direction. Sorry, I know I didn't answer your question, but.
I didn't really expect you to know what was with it.
Thanks for that. It's been a while since I've had more about depression. Can you speak a bit more to this remedial functional connectivity with biomarker?
Oh, yeah, of course.
Is it just being presented as sort of definitive phenomenon, or is there evidence for it?
Well, I mean, yeah. So well, there is a connection in the literature between this default mode network and depression in general. So, although I don't accept this idea of half the brain switching on and switching off when you're thinking and when you're executing a task, I think, well, as a complex kind of spaghetti picture that I showed is that all parts of the brain are involved in all parts of tasks. But there is a kind of very broad way of thinking that says, the more you internalize, the more you focus on the internal, the more depressed you are. Okay, so and that's what that hidden Markov model showed. And I mean, our research and that particular piece of work is not the only one that shows this connection with the DMN and depression. There's a lot, and there is work on this particular group of people where people were depressed following a traumatic brain injury, but this kind of pattern is shown in people with clinical depression who haven't had traumatic brain injury. So, I mean, the most common drugs that you give people with depression are SSRIs, right? So we thought, well, and psychedelic drugs are serotonergic drugs, of course, and we thought, of all these tiny nuclei in the brainstem, where can we look? And we looked at all of them, the ones we have ROIs for. There is a group in Harvard that has published a very comprehensive set of brainstem nuclei that relate to arousal systems, actually. And the arousal comes from our angle unconsciousness. That's why we're interested in those. So we tested all of those, and we thought it was interesting, but the only one we saw this kind of response was the RAFI, especially the median RAFI, which is heavily serotonergic. Is it an epiphenomenon? I mean, we don't know. Our data can talk to that yet. One thing that I can say is that we've replicated this with another data set. So at least it shows us, neither are published yet, but they're written up, so hopefully soon. So hopefully what it shows us is that it's not just a cortical phenomenon, right? And these modulatory relationships from the brainstem are important too. So that's as much as we can say at the moment. Although I'm sure Lenny is looking at me. Is there anything to add?
No, not necessarily. I mean, it's at the core, the idea of what depression might be is the more analytic hypothesis, like Emmanuel said, right? This is serotonin. So I think for a very long time we thought about depression drugs as things that, like you had today in the morning, have a locking key principle drug binds a receptor from there the effect is. Effect is typically been considered in very cortical terms. Instead, looking at the actual source of serotonin in the brain will give us an idea of why psychedelics sometimes just require 1 or 2 dosages to bring about quite significant therapeutic effect. It seems like this particular relationship, actually modulating the big scheme of the cortex, might be how this comes out. So what you saw in the beginning is basically when someone takes the drug, there's a sort of knockout effect, the connectivity disappears. After they've been treated, it becomes a lot stronger than it was before. And for that reason, there's this idea that potentially it is in a sort of hypo serotonergic state beforehand. Taking psychedelics, powerfully serotonergic drug, might reset how the serotonin system functions.
Is there anything that's one natural alternative solution? Is there anything specific that is
in depression?
Yeah.
Well, unfortunately, the brainstem nuclei for serotonin have basically been ignored for a very long time, other than animal research, which unfortunately we don't have many sort of behavioral reports. But it's definitely a very, very important question.
I mean, again, this was research that was sort of, we got into trying to validate what we've done in unconsciousness. I mean, is this the MRI scanners that we use vary in terms of strength of magnetic fields and stuff. And a 3 Tesla, which is it's a common kind of research scanner, MRI scanner, you can't see those nuclei. So there's a lot of people that say, well, this is all rubbish because you can't see the nuclei. But however, I don't accept that if I test all these other nuclei and I don't see any effect, and I see the serotonergic effect for the serotonergic drug, and the same we see an upregulation. If we test this, the ventral tegmental area with the dopaminergic drug, we see it upregulated. And again, with the same part of the cortex, posterior default mode network. And it's not a coincidence that you have, this upregulation of dopamine of the default mode network, and you have all this connection with consciousness and psychedelic drugs, especially posterior parts of the DMN. All these things cannot have just happened, right? Sorry, I didn't. I kind of went a bit fast on that part. So we're talking about 2 kinds of connections, and essentially 2 types of MRI imaging. One that is very sensitive to how the brain is actually physically connected, white matter bundles, as we call them. So you can do some complex, actually that particular technique studied here in Oxford. And you see how different parts of the brain, so you parcellate the brain to whatever resolution you want, to 5 parts, to 20 parts, to 1,000 parts, and you construct a matrix with the strength of the structural connectivity, of the strength of this bundle. How many lines go through the central point if you want? It's an example of calculating this. If you connect these 2 areas. So that's how you construct the structure, the scaffolding. So the thing that allows you to connect different brain areas. Now, functional connections don't have to be direct from a point A to point B. They can be the other points, right? So you can calculate the richness of the structure of the functional connection. Structural connection is rigid. What you see is what you get. That's it. It doesn't change unless you have disease that destroys some of this connection. So what we're comparing here, what we see here is if you're unconscious, the functional connectome and the structural connectome become very similar to each other. So there is no richness in function, right? When you've taken psychedelics, you see they move further apart. They're more different, the structural and functional connectomes. And less correlated. Yeah? Yeah. When you use correlations, there are a lot of other techniques you can use to find the same thing. You get an increase in small wellness as well. Yeah. I forgot to say, a lot of these papers are available here if you want to explore it a little bit more. I'm sorry, I can't.
Wondering what's the evidence all this kind of network?
I see. Well, there is another bunch of evidence that I didn't talk about, but there is. So if we try and localize in the brain. First of all, this is not research that we have done, but if we try and localize in the brain. Well, if you want social parts of the brain, the parts of the brain that help us interact with our environment, that allow me to answer these questions now, are heavily overlapping with what we call default mode network. So special areas in the inferior parietal cortex, in the medial prefrontal cortex, all seem to be important for us socializing. There are people in Harvard that believe, there's one particular researcher I have in mind called Rebecca Sachs, who believes the parietal part is more important. There are other people who give more importance to the frontal aspect of the default mode network in terms of being able to socialize. Now, somebody noticed this fact, that those areas overlapped with DMN areas. And I presume at some stage or other, this was a very, very early study. There are a lot more studies since then, which I'm afraid I haven't followed. But they said, let's check this and see whether there is any kind of correspondence. And that's what they found. The frontal aspect of the default mode network, in between the two hemispheres, if you think just about here, was differently connected in people who had autism than their healthy control, age-matched and demographically matched healthy controls. So that's what that slide was said. There's been a lot more research on that one since then, which I haven't followed, I'm afraid, but there's a lot more research out there on that topic.
So we talked a lot about kind of the connection between the research in anesthesia and kind of the source of consciousness, and that in psychedelics. And I was wondering if you could comment on this kind of more recent ideas that we may actually use psychedelics directly on people with source of consciousness, kind of bring them back to baseline. There's been a couple of papers on that. Though there seems to be kind of obvious ethical issues. So I'm wondering kind of what your thoughts on that are, whether you have any plans.
Well, it's quite interesting in that you see this continuum, so you think, well, how can I push this person who's down here up here, And I think Andrea has a good anecdote about that, because it was your paper, right? And we got a reviewer saying, you're unethical to suggest, we hadn't even suggested it, right? We didn't make a mention of that at all, but they said, you're unethical to suggest that you should give people with disorders of course the psychedelic. There is discussion going on. We're not involved in this, as far as I know, but there is discussion about that. And when I said, I didn't show a slide, but I mentioned earlier that we found a strong dopaminergic dysfunction in people with disorders of consciousness. I'm not arrogant enough to suggest that, brain injury is so heterogeneous. I'm not arrogant enough to suggest that that's the only thing that goes wrong in these people with severe injuries, right? Ideally, and one way to go forward with this is to build maybe connectomes for each of the brainstem nuclei, maybe connectons that are informed by the PET maps that Morten talked about earlier today, and bring or the small wellness or all that, and build a picture of what's wrong with each individual, and then decide on what treatment. Maybe more than one kind of treatment.
Just very briefly about the role of the relation to the people when they're I was wondering if any of you might perform that.
I mean, yeah. How long do we have? So, I mean, the posterior, well, both of these regions, the medial prefrontal cortex and the posterior parietal cortex, medial, are disproportionately connected to the rest of the brain. And there are areas that evolutionarily have expanded disproportionately more than any areas of the brain. The precuneus and posterior cingulate cortices have strong connections with the hippocampus. So in a way, and please don't quote these exact words that I'm about to utter, but I think, and we've seen all this evidence for integration of information. So I think they are, in a context of a predictive coding model, they provide the priors, right? What we know, what we have experienced. And I think then there are people who believe that there is some kind of comparison in the posterior part of the DMN. I think it's more the kind of comparative mechanisms that sort of update your priors happen more at the frontal cortex. But so you can have this kind of process where you have priors retrieved, and then you have evaluated, updated, priors retrieved again. So you can see how I'm thinking about this. There is a paper actually that is under review at the moment, but we have a preprint in our website, not this one, by Diane. And again, that shows that when you do a binocular rivalry task, the one where you're looking at two stimuli that are different color, and the stimulus doesn't change, however, you only perceive one color, right? And Diane showed in this other paper that uses both MRI and EEG data that were collected at the same time, that you see a signature of the change in the perception of whether you're next going to see red or green, or which stimulus, well, not which color, but the fact that a change is coming up in perception in the posterior part of the DMN. So before this happens, before you press the button to say, I've seen this, by perception change, you see a little signature of that in the posterior part of the deformal network. So all those bits and pieces make me kind of mention the DMN in the context of these predictive coding models.